Actually, my interest in cellular senescence started more than 30 years ago when I was a graduate student in the US. At that time, I worked with the primary mouse fibroblast. These cells are now called mesenchymal stromal cells. I use those cells to characterize, to analyze the somatic mutations that occur in vivo. So I need to get clones get the mutant clones from the fresh tissues. At that time, it's very hard. I mean, I could get the clones, but it's very hard to expand the clones. It's very hard to maintain. They quickly became senescent, so they stopped dividing. So they have limited proliferation potential. So and those cells look enlarged and flat. So that's a typical feature of senescent cells. Those cells, they look interesting, very interesting to me. But at that time, they are regarded as the end point of the lifespan of a cell. It's like at the end of their lifespan. At that time, maybe they attributed those cells to the aging process because of you have more senescent cells accumulating in the body. The body may aging quickly, something like that. That's kind of a recognition. In other words, maybe we regarded those cells as the innocent standbys. They stay there, but they other than they have functional loss, they may not do any harm to the body at that time. I think actually for those cells, I did some research on how the senescence occurs. We did some research on the signaling pathways by which gene damage causes cell senescence. I characterized several target genes downstream of p53. And that's very well studied factor in tumor suppression. Also, very important factor in driving cellular senescence. So I characterize several pathways like p53, CXCR2, p53, ROS feedback loop. So we studied those pathways that driving cellular senescence. I think two very important discoveries were made around 2010. That's greatly pushed the field forward. One discovery is that is the discovery of senescence associated secretory phenotype also called SASP. This phenotype can lead to tissue remodeling. Actually, nowadays we recognize those they also contribute to tissue remodeling in tumor microenvironment. So it's just those cells, not just the innocent standbys. They contribute to tissue remodeling. I think that's very important discovery. That's one of the important discovery. Another one is the paper published I think 2011 in Nature that showed that if the senescent cells are cleared, the clearance of senescent cells can delay aging associated disorders. That means if we can get rid of the senescent cells in the body, we can prevent or delay many of the diseases associated with aging. So these two discoveries indicate that learning to how to deal with the senescent cells is very fundamental to developing strategies that promote healthy aging and fight cancer. So I think these two discoveries reinforced my belief in this field. So we are continuing to work on this field. The RAD51 is a gene repair gene. It's very highly conserved gene repair gene. Actually, its homologue also occurs in bacteria. So it's a highly conserved gene repair gene. As I mentioned earlier, I studied mutations, somatic mutations. Mutations actually are caused by DNA damage. So at the beginning, they use various types of DNA damaging agents, like X-rays, like chemotherapeutics. Those agents can induce DNA damage, and those agents are also commonly used to induce cellular senescence. So if we treat, let's say we treat fibroblast or other type of cells with X-rays, the cells may enter senescence. So it's very quick models to get senescent cells. Another way, to get senescent cell is to pass the cells, passage after passage, like many cell doublings. You also get senescent cells. But really, like DNA damage agents can quickly give you senescent cells. So that's the way to get senescent cells. As I said earlier, gene repair function is very important for the repair of DNA damage. Actually, our gene repair function declines with aging. The older we get, our gene repair capacity decreases. And actually, many of the genetic disease called premature aging syndromes, like Werner syndrome, those people would look like 70s, even they are like 40s and 30 something. That's progeria. Many of those patients, they are caused by genetic defects in gene repair. So in the case of Werner syndrome, the gene mutated in that case is called WRN. So the gene, that mutation causes this premature phenotype. So because RAD51 is one of the most conserved gene repair genes, and it's very key player for homologous recombination, we generated the conditional RAD51 conditional mice in about 2014. Because RAD51 is very conservative, very important for development. We cannot study the constitutional knockout, because constitutional knockout mice, they die at an embryonic stage, like 3.5 with mice. So we have to study the function, the gene, the function of those genes. We need to generate conditional knockout mice. So in 2014, more than 10 years ago, we generated the knockout mice, RAD51 knockout mice, conditional knockout mice. So those mice enabled us to analyze the function of RAD51 in various types of tissues by using different types of clay. So in this study, we published in Hepatology, actually online last year, and officially this year, in 2025. In this study, we knocked out, we deleted the RAD51 in hepatocytes using tissue-specific using ARD crate. And we observed that in the knockout mice, in the hepatocyte-specific knockout mice, the hepatocytes quickly enter hyperpolyploidization. So normally, our somatic cells have 2 copies of the genome. They call it diploid. In this case, the one hepatocyte can have up to 16 or 32 copies of the genome. So I call them hyperpolyploid. So those cells are very, very large. And they quickly get into senescence. I think they get into senescence because they are polyploid state. They hard to maintain their usefulness, the vigor. Some they quickly get into senescence. Not only they get into senescence, they also create a chronic inflammatory environment. This environment is chronic inflammation. This chronic inflammation drives fibrosis and the tumor development, like in other chronic liver disease, like hepatitis, like NASH, that's non-alcoholic steatotic hepatitis, very common liver disease because of the increased obesity in the population. So I think this mouse model represents a very unique model system for the study of senescence-associated microenvironment. So with this mice, we can study how liver fibrosis occurs, how tumorigenesis is driven by microenvironment, and then also we can use this model to develop or to test the various types of interventional strategies like chemicals, like experimental drugs. This very unique model to study how we can modulate the inflammation associated with the cellular senescence in the liver. So it's a good model. So it will help to understand some of the problem, some of the questions in the clinic. So I think that's the meaning of this animal model. That's a question we keep thinking all the time. Because with this mouse model, I think we have the opportunity with this mouse model from the tumor development in this mouse model. We hypothesize that it's the senescent cells that create an environment that promote tumorigenesis. In this case, I think in the senescent livers, the senescent cells created a immunosuppressive microenvironment. So under this environment, the cancer cells would escape the immunosurveillance. Even the earliest, the earliest cancer at the early stage, if they escape the immunosurveillance, they will have opportunity to get more malignant. So I think this is the senescent microenvironment creates the tumor permissive. It's the tumor microenvironment, the senescent microenvironment creates tumor permissive microenvironment. So how, the question is how, that's kind of hypothesis. We want to test that. So is the microenvironment associated with the senescent cells immunosuppressive? We have to test that. We have to test whether this hypothesis is true. If it is true, we need to figure out how the immunosuppressive microenvironment is formed. What are the cells that constitute this microenvironment, and what are the functions of each of the cell subsets in this immunosuppressive microenvironment, and what are the signaling pathways that drives the formation of this immunosuppressive microenvironment. So those are the core questions we want to address in next few years. For this field of aging biology and the senescence, like cancer, the cancer development is also age associated with this disease. For this field, I think the importance, there's no doubt that this field is very important nowadays. Actually, it's getting more important as the society evolves in the future, because we live longer than ever before. Like in the I think in China, the life expectancy is around 80. So it will like several decades ago, it's not this high, right? So the aging population is the people over 60 or over 65. The population is getting bigger and bigger. So actually, the disease associated with aging will become more common and common. So to tackle those aging associated chronic disease, I think the aging biology and the regeneration medicine, they have a very important, play very important roles in combating those age associated disease. I think this field has very bright future because the demand we need to figure out how those age associated diseases occur, how we can develop strategies to slow down or to reduce the occurrence of those diseases. So that's a social need. From a personal perspectives, like for each of us, most of us, I think we have to deal with an old version of ourselves. Most people will expect to live beyond 70, 80, even 90, like Professor Yang Zhengning, who lived up to 103 years ago. So we have to deal version of old version of ourselves. We have to study aging biology and regeneration medicine is study our own biology. So I think that's something we need to. That should keep people interested and focused and fascinated. It's our each of us have it's our own interest. So that's my perspective for this field. And it's very important, and important to society, to science, also important for individual of us.